Dzień dobry Państwu, mam nadzieję, że mnie słychać. Marek Lorent-Kamiński z tej strony. Witam na drugim spotkaniu, drugiego dnia, czwartej edycji webinaru Wiedza ma warstwy. Spotykamy się pod koniec czerwca, żeby porozmawiać o nauce, biotechnologii i o tym, co słychać w przemyśle. Również dzisiaj spotkanie odbędzie się w języku angielskim, więc od razu uprzedzam. Jak zawsze w takiej formule 40-45 minut prelekcji, a później 15 mniej więcej minut sesji pytań i odpowiedzi. Proszę pytania zadawać na czacie lub podczas sesji Q&A można się połączyć na żywo i porozmawiać z prelegentami lub ze mną. Ok, w takim razie zaczynamy. Hello, I'm also welcoming you in English. Hello everyone, we're beginning the second meeting of the second day of the fourth edition of webinar series Knowledge Has Layers. Today we will talk about segmented flow, Droplet, Microfluidics and related applications. And today our guests are Simon Renard and Adam Mezien from Fluigent Deutschland. Hello, Simon. Hello.
Hello.
Hello, everyone.
Hi.
Hello.
Okay, so the floor is yours.
Yes, thank you very much. So we are both from Fluigent, Adam and myself, and I will start giving you a general introduction about our instrument for fluid management in microfluidics, and then my colleague Adam will take over. He is a product manager in charge of droplets at Fluigent. So let me start with my presentation. So as Marek just mentioned, I'm Simon, and I am the managing director of Fluigent Deutschland. So logically, we are based in Germany. We are a small team here of 7 people. In Jena, in the heart of Germany. That's the team in front of the first planetarium because they have been developed here in Jena. And since 2021, since the beginning of the year, we started to partner with Sygnis in order to sell our products in Poland and the Czech Republic. So, who is Fluigent? Fluigent is a French company based in Paris. We are about 55 people. That's a team here, so young people, I would say. And I highlighted this event because this weekend we will be present at the Élysée Palace in Paris. Showing our instruments to the President Macron because our instruments are wholly made in France. So that's something which will be celebrated. If it's not soccer, it has to be something else. Yes, and we have, as mentioned, one subsidy in Jena in Germany, but we also have one in the US, and we have worldwide distributors. Fluigent has been founded 15 years ago in Paris by a scientist, and today we have over 800 publications using our instruments. We have, I think, over 3,000 instruments in the field, and we are still innovative, and we are filing patents every year. Hopefully you will see the result very soon in our products. And we are focused on fluid management for microfluidics. So the disruptive idea to found Fluigent 15 years ago was to use pressure to control liquids for microfluidic applications. So many people use this gravity flow. You may have seen this kind of setup, homemade or bought at a company. And what we do is very similar, by pressurizing closed vessels, like for example this reservoir. And if we apply pressure, that's the same as if we would put very fast the reservoir at any point of this tower, for instance, with a very high resolution of less than 2 centimeters. If we take the 7-bar pressure range we have, and doing this within some milliseconds. And I think this explains the main advantages of pressure-driven flow. So we have no mechanical moved parts in the fluidic path, and everything is flowing very smooth. And yes, the advantage of having no pulsations for microfluidic are obvious. So this is how we are doing. We have here an instrument, a pressure controller with an air inlet here, and we have a gas outlet to the pressurized reservoir, and we are pushing out. Our piston is just the air. We are pushing out the liquid through the microfluidic setup. And if you compare this to a very high precision syringe pump, the best on the market, you would see that the pressure has a very stable flow. This is a blue line here. And with a syringe pump, you would see this these pulsations here, which are mainly due to this steep slick effect of the plunger on the syringe. So we have anyway stability which is below 0.1%, and that's really a great advantage if you have tiny channels. So, and one other advantage of using pressure is that you do not have any limitation on volume. So we could pressurize this kind of small reservoir like this one. So it's an Eppendorf, but it could be 15 milliliter, it could be 50, or it could be a large bottle. So we can adapt the size of the reservoir to the application. So it's contamination free, meaning everything which is wetted could be disposable, and we can parallelize different setups using one pressure channel. To show you how this stability is leading to a monodispersed droplet, I have selected here a short movie, which is if you see here the time scale, this is actually very fast, and you can see that every droplet has exactly the same size, even if it's you know, the flow is actually very high. So that's this advantage of having no pulsations. With syringe pumps, you would have maybe 5 to 6 or 7% of fluctuation in the flow. One other advantage of the pressure is that you would have a way faster response time than with other technologies, because you are pushing gas as long as you have not reached the pressure you want to have. So if you compare this again with a syringe pump, you would see this is a syringe pump, this is with pressure, and pressure is really very fast. Of course, this response time is depending on the size of the volume you have to pressurize. The larger the volume, the more gas has to flow in, so the more time it would take, but it would be anyway faster than with other pump types. So in order to illustrate this, I have here a few videos showing that you could, for instance, you can see here, this is a co-flow application with 3 different liquids, and you can very fast adjust the pressure, and you could also have a very stable flow profile. Yes, that's an example. One other example is here with a double emulsion. We can stop the flow almost immediately by pushing on the outlet, applying a counter pressure on the outlet. That's very fast, and that's something you wouldn't be able to do with another technology. And I have these are beads flowing through a channel, and you can stop the beads, and once they are stopped, in this application, we apply a magnetic field and could immobilize all the beads here. So that's a few examples to show you what are the advantage of pressure. So what do we offer at Fluigent? What is our portfolio? So we have basically, for end user, for the research, we have this kind of instruments, which are lab bench instruments to realize some microfluidic experiments. So we have pumps, we have valves, we have reservoirs, and we also offer different types of application packs. So we have, for instance, the Droplet line. This is Adam's baby, let me say this like this. And we have also all cell-based applications with our ARIA, for instance, or organ-on-chip applications, etc. And then we also have the ability to develop instruments for different customers. So we are doing this kind of integrated solutions. And we have an OEM product line where we supply bricks or elements to build fluid management inside an instrument. Okay, let me talk a little bit about our product line. So for the end user instruments, we have the fluid actuation with the pressure controllers, we have the flow rate measurement, and we have the valves, and we have a software, or we have different softwares actually. So we have a graphical user interface here. We have a script based software, a Fluigent script based software, and we supply also a software development kit where you could program or integrate our instrument in any of the automation platform like LabVIEW, MATLAB, or Python. So yeah, maybe I will stop sharing my screen and show you the instrument live. That's maybe slightly more interesting to see this. Can you see my camera? I don't, Marek, I don't see my picture here.
Yes, here and
Yeah,
no.
you
Actually, we see a black screen.
okay.
For some reason, you can try to reconnect the camera device and try to connect once more. Sorry for the technical problems. It happens.
Yes. Otherwise, I would stop share. Oh, I think that was a good. Yeah.
I had the same problem yesterday, and it was because my browser was collecting cookies, and it was connected with something like that. But I hope it's not the problem in your case.
Yeah, I can share my screen and do it with a PowerPoint presentation. That's not a problem.
Okay.
So back to this slide. So you can see my screen now, right?
Yes.
Okay. So basically, we have pressure controllers, which are able to control the pressure. But if you are working only with the pressure, you're kind of blind because you do not know what happens with the flow rate. So we have some flow rate sensors we can place into the fluidic line here so that we can measure the flow rate. So we have a kind of control of what happens in the fluidic path, and we are able also to control the pressure in order to have the set flow rate. So we can use the instruments in two modes. One would be the pressure mode, and the other one would be the flow rate control mode. So you can just set the flow rate. And this is an example for cell culture here. If you have long-term experiments, maybe the hydrodynamic resistance, maybe you would have a bubble or some particles inside the tubing, and then the pressure is adjusted because we know the flow rate, so that the flow rate remains constant. So that's an example. So we also have a different valves. So we have a two system to control the valves, the switch easy, which is compatible, which could be clicked with the flow easy modules. And we also have the switchboard, and then you could control this kind of three-way three ports two-way valves, or this injection valve, or we have also a rotary valve here, which has 10 inlets and 1 outlet, or the other way around. So this is an example of what you could do with this rotary valve. You could have only one pressure channel, and with a manifold, dispatch the pressure to different reservoirs here. And by selecting the outlet with this rotation, you would be able to inject up to 10 different liquids sequentially through the system. And that's of course an advantage of pressure, because you can split the different pneumatic ways. So that's an example of what we could do with our software. We can also program different pressure or flow profiles, which may be used for cell culture applications if you want to be very physiological and mimic the blood pressure, for instance. And we have here in our automation tool all the different instruments, and you can by drag and drop just select the different actions you aim to do. That's an example of what we could do with our to select different beads with either with a software development kit or with a TTL control we have on the link module. So you would be able to sort the different particles with our software with these two different possibilities. So I have shown you this rotary valve, which is the core of this instrument called ARIA. ARIA is for cell culture applications, or for microscopy, for all kind of special omics or immunostaining, because you could inject up to 10 different liquids into a microfluidic cell. And this is basically the same hardware than we have with the end user components. But here we have integrated a very easy to use software. We just have to enter the sequence of liquids you would like to have and just press start. This here is an example of what we could do for customers. These are custom systems. So you see here 3 rotary valves in different reservoirs of different size. That was a project for a hospital. This is a very nice design instrument for the cosmetic industry. So the design seems here to be important. And we also have this kind of specific platform for specific requirements. In that case, it was for cell encapsulation in droplets. Yes, and then we have, of course, our OEM components. So we have different pressure controllers. We have this one called the POEM. So this is could be just screwed inside the different instruments. We have a single module here called PX, which is very robust, could be used as a RS-232 or USB instrument. And we also develop some further instruments for the customer needs, or we just deliver some part of the fluid management. So the assembly of all our instruments is made in Paris, and we are able to deliver a few units as well as a large amount of instruments. So we can have this scale-up possibilities in-house. Yes, and I think for my part, that's it, I would say, and I would give the word to Adam. And let you talk about the droplets.
Yes, I will briefly try to share my screen. Can you see my screen and can you hear me well?
Yes.
Yes? Okay, perfect. So then we will start. Yeah, just to say some word about me briefly, as Simon said and Marek, my name is Adam Mesian. I'm working as project manager at Fluident, and my work is focused on really droplet-based microfluidics, how we can use this method for different applications, and the development also of complete solution for different applications, which involves the use of droplet-based microfluidic. So first of all, I would like to go back a bit to the context, which is the generation of emulsion. What is the emulsion and how do we do it and what are the applications? So basically, an emulsion is a mixture of two or more liquids that are immiscible. So as we can see here, so maybe I will use this here. It's quite easy to make emulsion. So the idea is to have a reservoir which will be filled with two immiscible fluids. For example, it could be oil and water. And we are going to use forces. It could be mechanical, like this stirrer, but it could be also ultrasonic forces. And using this method will allow us to generate emulsion. Kind of the same idea that you are using when you are making, for example, mayonnaise or vinaigrette. This is the same idea, actually. And this will lead to an emulsion, so the operation of droplets inside a reservoir which will be filled with two liquids. So basically, there is two types of emulsion. I said types because actually there is different solution, but emulsion could be water in oil phase, so such as aqueous solution or hydrogel solution, or oil in water, such as polymer, mineral oil, fluor, or carbon oil. But it could be also water in oil in water droplets and more complex emulsion. But this is another point. So actually, the emulsion has many advantages for many applications. And this is a powerful technique which allows to, for example, make cream in cosmetics. For example, encapsulate inside droplets caffeine for anti-aging cream production, for example, or for fragrance manufacturing to control the slow release of a flavor. In food also, to encapsulate, for example, the vitamin inside droplets, and also for test masking, to have the encapsulation of, for example, a drug which would have, let's say, a bad taste, and to encapsulate in a phase which will be more acceptable. Also, in pharma is a big also market for particle and droplets. So the idea is to have an encapsulation of drug inside microparticle or microcapsule, and by controlling the size and the shape of the microparticle, you can control the release of a drug. You can protect also the drug during physiological transport to have targeted drug delivery. So to make sure that your particles, your drug delivery system will target a specific organ, for example. And also in research, when you want to encapsulate cell at single cell level to have really an identification of a single cell, and also to make next generation sequencing, or to make other tests for cancer, drug testing, for example. So this is, let's say, an overview of all the application that can be where the where droplet and emulsion are used. But how do we make emulsion exactly? So there is many methods to make emulsion. These are some of them: spray drying, jet milling, or extrusion. But the most common method is the emulsion in batch, as I presented before. So this is exactly what I presented. So 2 immiscible solutions in a reservoir, and you will generate forces. So the advantage of this technology is that you can generate a large-scale production from kilogram to ton per day, for example. This is something which is conventional, which is widely used in industries, and which is FDA approved, FDA, Food and Drug Administration, for the development of drugs and other drugs like this. However, as we can see here, there is many limitations of this technology. The biggest limitation is the polydispersity, which is the fact that you will have many droplets which will not have the same size. When you are making that kind of experiment in different markets, you want to target one specific size, because a size will be directly related to a drug release, an encapsulation efficiency, and so on. So the idea is to have only the same size. So when you are making an experiment like this and you have this result, you will need to post-treat your sample and get rid of all the droplets that you are not that are not the same size. So you are wasting reagents, money, and also time, because you need to add the post-treatment. You do not have any control on microparticule or droplets size. So you have different size. You have a different size, so you're not controlling the specific size. You have also a long encapsulation efficiency. You cannot control the encapsulation efficiency when you're making droplet. You will have, for example, cells around the droplet and not all the time inside the droplet. And also, which is important, is that using that kind of forces, you generate stress on encapsulated agent that could modify or damage the solution. A droplet-based microfluidic appears as a solution to this. The idea is to handle 2 immiscible fluid, again, but at micrometric scale to generate micro droplets, as Simon presented. So here's the kind of setup that we can have. So here we have fluid handling system, so the pressure controller that Simon presented, an oil here, water, and by controlling precisely the flow rate, so here we have flow sensor. So we are going to regulate the pressure to control the flow rate. And then we are going to use a microfluidic chip to generate droplets. By controlling this flow rate precisely, we will be able to generate really high monodispersed droplets, as we can see here. So the advantages of this technology is that we have better control over the process. We increase drastically the monodispersity. As you can see, we have always the same size. And also we can play with different size of droplets. So at the beginning, the droplets were bigger. Now they are a little bit smaller. So this is on purpose. And when you are doing this, you have up to 95% encapsulation efficiency. That means that you're not wasting any agents. You encapsulate everything. However, the limitation are, which is something which is a less known method, it is really adaptable to lab scale production for R&D, for researcher. However, it is not suitable for the moment for large scale production, which are kilogram or ton per day. So this is the big challenge of the microfluidic. Here, as you can see, we can change also the frequency of generation. So why are we using these droplets? Actually, one droplet equals 1 microreactor where chemical, biological reaction can take place. So as we are in the miniaturization, we have small sample volume, which will lead to cost decrease. Also, compartmentalization, which leads to better sensitivity. You have a less diluted solution here, so you will have better sensitivity when you are making experiments. For example, fluorescence experiments, which is important. Short time for reaction and analysis, as we are using picoliter micro droplets. You will have a shorter time for reaction. Rapid stability of temperature, this is the same. We have better diffusion. And also automation and integration are possible. For example, is a kind of a nice example here. We are calling lab on chip. So the idea is that, for example, you can encapsulate a cell inside a droplet, and by controlling the flow, you can say that your droplet will go from a point or another point where chemical reaction can take place. And this can replace actually what a technician would do over a day. Here, that can be done, for example, in an hour inside a machine, for example. So a brief example, this is from an article, for example, for the same total reaction that we would like to do, a comparison with a pipetting robot and a microfluidic droplet. Total time, for example, here will be for the robot 2 years, here it will be 7 hours. You will be able to generate more reaction per day than the robot. And in terms of costs, if you translate it, using microfluidic droplet will be more cheap. If we compare, for example, the traditional batch method that I presented, we have a dispersion in terms of size, which is around 20% in the best case. With microfluidic based method, we can go down to 5% and below below 5% actually. So here we can see that we have really high monodisperse droplet anytime. With a batch method, you can see that droplets are not, they don't have the same shape and they do not have the same size, which is the big limitation. Okay, now I propose you to present you what is the Fluidian solution that we are proposing for droplet-based microfluidic. So we are providing a versatile tool for emulsion production and droplet production, which is based on the Raydrop, which is this device. And also, this is a combination of this device and also all Fluidian equipment, pressure controller, flow sensor, switches that we are going to automate, that we can automate, that we are going to integrate in a setup, in a package for complete solution to generate droplets. What is the Raydrop? So this is a microfluidic device which allows to produce highly monodispersed droplets for many applications. So the idea here, I will not go in detail, but the idea just to present you is to have a stainless steel device here, which will contain a chamber of a volume of 2-3 milliliter. And this chamber which will be filled with what we call the continuous phase. For example, it could be oil. And if you want to make water in oil droplets, we are going to have a dispersed phase, which will be the water. We are going to inject water inside an ejection capillary composed of a nozzle here, aligned with an extraction capillary. And by controlling the flow rate of the continuous phase and also the dispersed phase, we will have a squeeze, a shear stress in all dimensions of the capillary, which will lead to a generation of highly monodispersed droplets. So the Raydrop is composed of 3 main elements: injection capillary with a nozzle, extraction capillary, and also a body. Everything is exchangeable. With this technology, we are able to emulsify any type of liquid. We can make water in oil emulsion, such as water in mineral oil. We can make water in fluorinated oil. This is a bit different in terms of viscosity and surface tension. And also oil in water emulsion. And this can be done in the same device. So that means that you do not need to change your device to make one application or the other. One Raydrop device can do actually the 3 application. The benefits compared to other microfluidic chips, this is that you do not need any coating. When you are making a droplet with other microfluidic chip, every microfluidic chip needs a coating specific for her application, let's say. If it's water in oil, oil in water, the coating will be different for the microfluidic chip. Here, we can perform any type of emulsion, water in oil or oil in water, with the same device. You do not need to add any surfactant for droplet generation. However, if you want to recover it to have a stable emulsion at the end, you need to add it. We can generate highly monodispersed droplets. Dripping regime is a regime where you will generate highly monodispersed droplet. This is something that we want to have. And also high consistency. So that means that the flow rate variation, small variation, does not impact the droplet production. Now I would like to go a little bit more on the application and what the Raydrop can do actually. So because the Raydrop is versatile as I presented, it can cover many applications such as polymer microparticle production. The idea will be to generate polymer microparticle, first polymer droplets, which will contain, for example, drugs. This polymer can be FDA approved, and for example, PLGA is a FDA approved biodegradable polymer. It is used for encapsulation of active pharmaceutical ingredient, and it allows to generate emulsion, which will be tunable and can deliver for a control time the active ingredients of the drug into a patient. And using this method, we can generate that kind of particles with a highly monodispersity, as we can see here. For example, a test case were to use a lysozyme, which is a protein. We encapsulated the lysozyme inside PLGA microparticle. So we were making droplets, and after that, there were a chemical reaction, which will lead to microparticle production. And we have tested different concentration of PLGA, for example, different concentration of lysozyme. And we can play with droplet size from 50 to 100, which will lead after that to a particle which will be smaller, 30 to 12 to 30 micrometer. Dispersion lower than 3%, as I said before. And we have an encapsulation efficiency which will be higher than 95%. That means that 95% of the encapsulated agent is recovered inside the microparticle. We are not wasting only 5%, let's say, agents. Other application is hydrogel microbit production. So the idea is to use again the same setup, the same solution to generate droplets with a specific hydrogel. And this hydrogel is widely used for pharma, for drug formulation, in research, for cell and protein culture inside microbeads, in cosmetic for toothpaste or fragrance manufacturing, and also in food for encapsulation of vitamin inside microbeads. One example is an encapsulation of organoid inside the hydrogel beads. And then we put this hydrogel bead into culture. And after that, we were able to grow the organoid to make some experiment after that, such as transfection. This is something which can be done also. Okay, that was for the alginate. Here is another version of the Raydrop. So this is based on the same device actually with a small modification, which allows to make not single emission, but double emission. So the idea, I don't know if you can see here, is to make one droplet inside another droplet. And this is something which is really interesting in different area to make microcapsule for drug formulation. When you have drugs which are hydrophilic, hydrophobic, so different type of drugs, you will need to sometimes have a double emulsion for cosmetic also, and in food for test masking. So the idea here is to, as the previous Raydrop, so this is the same idea. So we will have two capillary aligned in a chamber, which will be filled with a continuous phase. The only difference here is that here in the nozzle, we will have actually two nozzles. One is for the core phase and one is for the shell phase. So the core phase, which will be this solution, and the shell phase, which will be the outer solution. The idea here is that we are going to generate double emulsion like this using this alignment of two capillary. As we do not need any coating to make any type of emulsion, here this device is totally suitable, and this is the only device available on the market which can generate robust double emulsion production. So here I will present you a small video. Here's the setup we use for the video. So here the free pressure controller, which will be connected to a core phase, a shell phase, and a continuous phase. That was water, oil, and water. There are 3 flow sensors. These are switches to automate the priming of the system to make sure that you do not have any air bubble inside the system. And then the Raydrop to generate double emulsion. So here is a presentation of the workshop that we did focused on double emission. Here you can see the AIO software, which can control the flow rate and pressure. So here we have the pressure controller for the pressure control for the continuous phase, the flow rate related to the continuous phase. Here is the pressure for the shell phase, the flow rate for the shell phase, and pressure for the core phase and flow rate for the core phase. Here we have a visualization of the user. Here is the platform that we used as I presented before. And here is the visualization of the microscope, so the Raydrop, what we can see inside the Raydrop. So the idea is to first inject some pressure, and which will lead to an increase to the flow rate of the continuous phase. So here you do not see anything, but you have actually a flow of the continuous phase, so the phase in the chamber, which will go out inside the collecting insert. Sometimes you can see some dust moving. We are going to increase the flow pressure of the shell phase to inject first the shell phase and make droplet of the shell phase. So nothing is happening now. We need to switch a valve to inject the shell phase. Here is what you can see here. So it takes time, so we are going to increase a bit the pressure to increase the flow rate. So we just maybe go a little bit quickly here. And here we are generating a flow on the shell phase, only on the shell phase. When you have a smooth, let's say a steady state for the shell phase, you see that your flow is nice. You can decrease the flow rate of the shell phase, the pressure of the flow rate and the flow rate of the shell phase, which will lead to a droplet formation. And then we are going to increase the core phase to generate flow rate of the core phase. And we are switching the valve to generate double emission. So this is the idea. It's quite simple, actually. As you can see here, there is a small instability that appears, which is mainly owing to the flow rate. So it is important when you are making double emission and single emission to have a stable flow rate. As we were at a transition regime at the beginning, we have a small fluctuation, and which will lead to instability. So you see that having a flow rate, constant flow rate, is really important. We have changed the pressure on the shell phase, on the core phase. Sorry, we are going to decrease a bit. And when we have a stable flow rate, now we are in a production regime, and we can generate the droplet for a long period of time. You can see that the flow rate is stable, and this is the idea. So after that, we can generate droplets, and I will go a little bit later. And then you can collect your double emission, and here is the kind of double emission that you can recover. So this is the idea, and everything is working really well here. Okay, we'll go back to the presentation. Using this method allows to produce, again, double emission with any type of solution, water in oil in water double emission, as we can see here, for different type of oil, a surfactant that we are fluorinated oil with a surfactant that we are proposing, a mineral oil. Here, the opposite with the same device, mineral oil in water and mineral oil. And here is for partially miscible double emission, such as solvent, water in HA acetate. And as you can see, controlling the flow rate impact the shell thickness. This is really important. That's why when you're making emulsion and double emulsion, you need to have a stable flow rate. And that's why we are proposing pressure-based flow controller. And we can play with the size of the double emission from 80 to 150 and again more. And also core size between 20 and 120 with the same monodispersity. So always having lower than 2% polydispersity. There is also another application that the Raydrop can cover. This is for nanoparticle production. And here we have developed a package for liposome nanoparticle production, which is something which is widely used for cancer treatment, production of drug delivery system, also in cosmetic. And it's something which is really important and actually in the current time, because it's widely used for vaccine development. And for example, the COVID vaccine, which has been developed by Pfizer and BioNTech, and also Moderna, are based on mRNA encapsulation in liposome nanoparticles. So here's something that I wanted to show. because it's a bit different than Droplet. Here, as you can see, we are not making droplets. This is a co-flow, we call co-flow. So the idea is to mix a inner phase, so the dispersed phase, and the continuous phase together. And we are going at the end to mix the two solutions, which will lead to, if we are using the good chemical solution, which will lead to a nanoprecipitation and nanoparticle production. So we are not at micrometric level, we are nanometric level. And we are able to, by playing again with the flow rate, so this is here the flow rate ratios, this is the ratio for the flow rate of the dispersed phase and the flow rate of the continuous phase. So this phase here and this phase inside the nozzle. And by playing with this flow rate ratio, we can play with the size of the nanoparticle. And this is important. So having a stable flow rate is again important. And we are able to generate nanoparticle size from 30 to 180 nanometer. Here, we are not at droplet microfluidics, so polydispersity is a bit higher. But actually, when you are making nanoparticle, the polydispersity of, for example, lower than 50% is really a good number. I don't know if I still have a lot of time, but I will go on other application. So we can propose also briefly some application. So here is an application. We are proposing the application package to make single cell encapsulation and study the single cell level. So the idea is to encapsulate one bead and one cell inside the droplets to improve current genome sequencing techniques. And the method is called the Droplet method, which is also widely used. And the Droplet method has been highlighted in 2015. And we are proposing this solution to generate the emulsion and have encapsulation of single cell and single bead inside droplets. Other application that we can cover also is to encapsulate cells inside droplets, aqueous droplets, for example, with media, and to make cell growth. I don't know if you can see well here, but you see that some cells are containing here, some droplets are containing cells, some are not containing. And after that, if we inject it in the, for example, sample, which will be in incubator, we can see that you have cell growth on some droplet and nothing on the other. This is also widely used. This is a good technique for researcher. And also another application, it's digital PCR for diagnostic. So the idea is to have amplification of DNA for different diagnostic. For example, COVID diagnostic is based on PCR. Here, the idea is to make digital PCR. So we are going to use a sample with a DNA, generate emulsion with this sample to have droplet which will contain DNA, then amplify it, and have after that droplets have better sensitivity and have some droplet which will contain the amplified DNA and some which will not contain it. And we can also propose the complete solution for that. So as a conclusion, I would say that droplet-based microfluidics offer better control and precision for emulsion production. And this is really powerful for encapsulation of active ingredients and living cells. The big challenge of microfluidics remains for large-scale production, but this is for industries, and this is where the microfluidics is going now. So we are moving to that. And we can propose at Phrygians complete solution and method for many range of application, such as I presented, polymer microparticle, microcapsule, single cell encapsulation, or cell culture inside droplets. So that's all for the presentation. Thank you for your attention, and I think it's time for discussion and question.
Yes, thank you, Adam. Thank you, Simon. That was really great. And actually, we have the first question from Marek Brzeziński. What kind of solvents can be used for nanoparticles preparation?
That's a good question, actually. Basically, the Raydrop was based on a nozzle which is metacrylate, made of metacrylate resin. So we have tested different solvents. Maybe I have a slide actually for that. So we have tested many solvents such as ethyl acetate, chloroform, dichloromethane. We have seen that with the metacrilat nozzle, dichloromethane and, for example, chloroform and DMSO had impact, damage a bit the nozzle for a long period of time. That's why now we are going to propose a glass nozzle. So based on that, the glass nozzle are compatible with kind of every solvent. For your question regarding nanoparticle preparation on the liposome, we used acetone, and acetone is compatible with the metacrilat nozzle and also the glass nozzle.
Great, thank you. Okay, and as for the nozzles, I would like to ask, how do you replace or clean the clogged nozzles?
Actually, that's a good question, because cleaning is kind of, let's say, kind of issue when you are making when you are working at microfluidic level with microfluidic chip. It can be a little bit tricky to clean some nozzle. Here we have a solution, as I presented, which is really based. I don't know if you can see my screen still.
No, you're not screen sharing right now.
I will.
Oh, yeah.
Yeah, perfect. I will go back to this. Here, the idea of our solution is that the user can have a flexibility on this application. And if a nozzle is clogged or an application goes wrong, for example, the idea is that he can clean the nozzle easily. So we have different protocol based on the use of solvent, for example, or injecting continuous phase. For example, clog the droplet collection, clog the continuous phase here, and inject the continuous phase at a high pressure to have a backflow of the flow here at the nozzle to remove the dust, remove the particle which is clogged. But also, with something which is important is that we can put the Raydrop in ultrasonic bath, filled with a solvent, for example, and let it like this to remove the clog. And if this is not working, we can also go a little bit deeper. This is something which we can do here. This is removing, as I said, everything is extendable. So we can remove this insert, which contain the nozzle, this insert, which contain the extraction capillary, use it, try to clean it with a syringe, with a solvent, for example. And you can actually clean it really well. So there is no problem for that. There is mini protocol and you can clean it well. If at some point you have made an application where everything is clogged and you cannot clean it, the advantage of this technology is that you do not need to buy another Raydrop. You just need to change this nozzle or change this capillary, buy another one, and we'll send you another nozzle or a capillary that you will directly connect inside the body of the Raydrop, and you can do it by your own. So you do not need any support or something like this.
Okay, great that it's intuitive also to replace the parts by yourself. Okay, and as for the Raydrop, is there any wear over time? What's the lifetime of a device?
So as I said, actually, it depends on the application. For example, so now we have different nozzle for the Raydrop. So some solvents are not compatible with the metacrilat nozzle that I presented. So if you are using a solvent which is not compatible, you will damage the nozzle. So this will have, let's say, a small lifetime. However, for other, if the solvent is compatible, for example, if you're using glass, actually you do not have any lifetime regarding that. It's just based on the compatibility of the solution that you're going to use.
Okay. Yeah. Okay. Thank you. So if you have, if our guests have any questions, feel free to ask. We have like few minutes till we'll have to disconnect because we'll have another webinar coming soon. But I would like to ask one last question about the advantages of your technology. So what are the advantages of your system comparing to other microfluidic methods for droplet productions?
That's a good question, actually. So there is what I presented. So based on the, let's say, focus on the microfluidic device for the droplet production, there is the Raydrop, which is something which is versatile. You do not need, so everything is based on the geometry. So that means that you can make any type of emulsion. When you are working on microfluidic, droplet microfluidic, usually you are using one chip for one specific application. If you want to make another type of application with another solution, for example, if you want to make water in oil or oil in water, you will need two different chips. Or you will need one chip which will be coated, but every coating has a lifetime, and this is not something, a solution which is robust. With the Raydrop, you can emulsify any type of liquid, which are compatible with the nozzle, without changing anything, without coating needed. So this is the main advantage of this technology, and this is why it's really useful for double emulsion production. So this is
if you need different droplet sizes, we have different nozzle pairs, so or capillary pairs. So that means that you would have for small droplets smaller diameters, for larger droplets larger diameters. So you can use this body for a lot of different types and size of droplets.
And are they coming with the systems? Are these size of the different size of the nozzles are coming with the systems that I bought?
You have to configure it.
Configure, okay.
Yeah. You say what is a droplet size you want to achieve.
Okay, for specific solution.
Yeah,
exactly.
The idea is that you say that you want to achieve a specific size of a droplet with your liquid, and we can propose you different size of nozzle and capillary. And also, if you say that you do not know, you want to have a big range of droplets, you can have one specific configuration, but you can also have additional nozzle or additional capillary, where you will be able to change it and to target other application, other droplet size.
Okay, yeah, I get it. Great. Okay, so I guess there are no more questions from our visitors. Thank you very much for the presentation. It was really great to hear about Raydrop and other solutions from Fluigent. Thank you, Adam. Thank you, Simon.
Thank you.
That was really great. And I would like to also welcome you to maybe stay for our next webinar. We'll talk about 3D printing and printing with from wax, actually, at 12:30 a.m. Dziękujemy Państwu.